Cosy Science. So it's Ale at the camera, I'm Natalie, this is Michael, and this is Mario. And we are a group of young scientists, for those who do not know, that are committed to communicate science with high-profile scientists to a non-scientific and scientific audience, so that everyone has access to scientific topics and everyone is allowed to ask questions. So we are today looking forward to talk about cancer, and Mike is going to introduce a little bit afterwards, but I'm going to explain how the evening is going to work. So everyone, I hope, has got tickets for a free drink. If not, you can come later and get one. Professor Pellman is going to talk about roughly 20-30 minutes, and then we will have a bit of nibbles and another drink, and Mario is going to explain the game. So we have a game, which is very difficult. We always have that in the break. So the break is going to be 15 to 20 minutes, and I'm sure you're not going to be bored. Afterwards, we open the question and answer session, which is usually the most interesting part of the talk, and I hope everyone is enjoying the evening.
I'd like to introduce our sponsors, which is the Company of Biologists. And they're a non-profit publisher and a UK charity, and they've actually provided the drinks and food, which I'm sure you're really loving at the moment. They publish 3 internationally acclaimed journals, which are Development, Journal of Cell Science, Journal of Experimental Biology, and 2 open access journals, which are Disease Models and Mechanisms and Biology Open. The money they raise is used to provide grants and sponsorship for innovative research, scientific workshops, and our company work. So if you have any questions about this, then about the Company of Biologists, then you can speak to Sarah or Catherine, who are here. So our speaker today is from the US, from Boston, and is Professor David Pellman, and he's currently the Margaret Dyson Professor of Pediatric Oncology at the Dana-Farber Cancer Institute. He's also a Professor of Cell Biology at Harvard Medical School and an investigator at the Howard Hughes Medical Institute. His lab studies the mechanisms of underlying normal cell division and cell division defects in cancer cells and cause genetic instability. And today he'll talk to us a little bit about evolution of cancer.
Okay. Actually, thank you so much for having me. This is called Cosy Science, and actually, I feel I've never done this before, I have a little bit of thinking to do of how best to approach you guys. Now, first, I was misled, I should say. I was told that it was a strictly non-scientific audience, but I think I can smell a graduate student like a mile away. So how many graduate students and postdocs are there here? Okay. This is bad. So I actually have to be accurate, which is really. I was hoping I'd have the gifts of poetic license. I'm going to talk a little bit about the science that I do, and I'll tell a story about something that I'm interested in. But in thinking about what might be interesting for the general public, well, I'm interested in how cancer genomes evolve, how cancer genomes get to be different from normal genomes, and if we can learn something about that, can we find good and better ways to kill cancer cells, which is a tough problem, right? Because the cancer cells come from the normal cells. But sort of in thinking about what to put together here, I realized that a lot of people are interested in biology from a very personal perspective. I mean, thinking about biology is kind of thinking about who am I and what really am I. And I thought, that which of course got me thinking about me. And I thought that it might be worth, as I sort of thought over my career, so I've lived through the whole genome revolution, and I thought it would be interesting just to hit a couple of high points, not of my career specifically, but just to show you and people who are outside the field how all the new genomic information changed my career and totally changed the way I do science. At the end, so I've taken on a new job. I have a friend of mine who's head of a nonprofit genomics institute outside of Philadelphia called the Coriell Institute. They're taking on a new project which he's wrote me into, which is they're trying to set up personalized genomics. Has anybody heard about this? So there are companies out there that want your money. So they want, the wife of one of the Google founders has founded this thing called 23andMe, and they want you to sign up and get your genome sequenced. And my buddy Mike Chrisman has set up as a nonprofit to try to genotype and ultimately sequence large populations. And he's roped me into trying to evaluate how do we use that information in an interesting way. So I thought that at the end of the talk it might be interesting to think a little bit from a personal perspective, how we are all, and certainly our kids, are going to really use genomic information in our daily lives. So I had a couple of questions that I thought I'd throw out for discussion. You can kind of think a little bit about them at the talk. Maybe we can discuss them at the end. So question number one, if you knew, this applies mostly to women, but imagine it could apply to men, that you had a mutation that gave you essentially 100% chance of getting breast cancer by the time you're 40. And not just getting breast cancer, there are different kinds. This would be a particularly nasty kind of breast cancer. Would you want to know? So that's an interesting question. And then the second question that I think is interesting to think about is, would you give some company or our nonprofit samples of your DNA to get the sequence of your DNA, so that as medical information comes in, that you could learn stuff, that instead of having 100% chance of getting this, that, or the other thing, that you might have 2% chance? And would you want to know that? So I think those are some interesting questions. So, okay, so we're going to start at the beginning. We can't talk about genomes without a little bit of definition of terms. So we have to say what's maybe I can get some help from some of the students. So can somebody want to define what a chromosome is for your compatriots here that don't know obviously what a chromosome is?
It's condensed DNA. It's DNA condensed around chromosome. Right.
Exactly so. And is there somebody in the audience here who doesn't feel any smarter now? Right. So the DNA has, it has a code. It has information. Everybody has 46 chromosomes. 23 come from mom. 23 come from dad. That's why the company, the Google founded why this company is called 23andMe. And chromosomes, so you get them, they're in human genome, there are about 3 billion bases, that's individual parts of the code, sort of like for the computer people, the bits. 3 billion used to seem like a really big number until in the US we started hearing about how much money we owe to China. Now it seems like a kind of manageable sort of number. Okay, so every cell, we have billions of cells in our body, and each one has a pretty much exact copy. I think pretty big. Well, at first pass, you could say that that's a pretty exact copy of of the chromosomes. So where did that concept of chromosomes come from? It's not the idea that the chromosomes actually carry information that make you and me different is actually not that old an idea. The way that was discovered, I think, is interesting to talk about in a few seconds, because the way it was discovered sparked things that are interesting to me and things that I'm working on in my own lab. So there are several people who would lay claim to discovering that chromosomes carry the DNA. I think the most important guy was a guy named Theodore Bolveri, and one of the beautiful ways that he did this was just by looking. So this is, I think, to me, I'm a cell biologist, and cell biologists essentially look at cells. And I think that this series of experiments really illustrates the power of just looking. Actually, you can't just look; you have to look and think. But the looking is the sort of the proximal part of the equation. So, he watched cells dividing, and people had not in real time. He was taking fixed cells, people had just developed dyes so that you could see the DNA and you could see the chromosomes, but nobody knew what they were. Nobody knew what was in them. Bolveri was studying oocytes that were really big egg cells, and he could fertilize them. And he could, so I told you one sperm comes from dad and the oocyte, the egg comes from mom. So he created a kind of menage a trois where he got two or three sperm cells into the egg, which created a lot of havoc, which you might think would be a very confusing experiment. But it wasn't a completely confusing experiment. What happened is that he watched carefully, and he noticed that these chromosome things, these dark blobs that you can see in the microscope, got separated in more random ways when you introduce this funny menage a trois. And then he kept looking. That was actually the smart part. So when the egg develops and gets fertilized, it goes through a kind of stereotypic pattern of where the one cell ultimately becomes a skin cell, and this becomes a bone cell, and that becomes a brain cell. It's not in different organisms. It's more how shall I say, deterministic than in others. And he looked at what came out, and what he noticed is that when the numbers of chromosomes were all messed up, then the fate of the cells was really altered. So that was actually the brilliant connection, just from looking. This is like the pre-Socratic saying, well, it would make sense if there were atoms out there. So he hypothesized and then went on. Actually, there's a lot of biologists in the audience now. A very fashionable thing in biology now is to have collaboration between physicists and biology, and to use mathematical algorithms, right, to try to get a better and more precise understanding of biological phenomena. That's an exciting and interesting approach. But it's not very novel, because actually Bolveri had his own physics friends, and they actually did a lot of statistical modeling. So even back in this, he was looking, but he was also thinking too. And so they were able to not just say, when you've got an abnormal number of chromosomes, that you've got, funny pattern of development, but they could actually make some quantitative statement about the probabilities of what the developmental outcome would be with different numbers of chromosomes. So that's the discovery of the chromosome. Bolveri also wrote a really marvelous monograph called On the Origin of Malignant Tumors. And that's for scientists